Good morning, everybody. So I've been traveling a little bit, and in the lead up to this meeting, there was a conference call to work out how this session would go, and I missed the call. And then I read the minutes after the call and found out that I would get up last, and I would talk about how we were going to solve HTE in RCTs, and I would do it in 15 minutes. So I'm going to take some very broad swaths in this talk, and I'm probably going to say things that are so summarizing as to probably offend at least one person at every table. But let me try to march through. I also thought about starting off with a slide of climbing in Scotland, where you get up to the top of the mountain and you can see everything clearly. And that is ideally where we want to get. We want to have some sense of exquisitely the exact therapy that the patient would absolutely want, be most likely to benefit from. However, we look out over this cloud inversion, and every valley around us is filled with clouds, and as soon as we walk off the top of the mountain, we end up in a very unique valley filled with cloud, and everyone tries to solve the problem for navigating inside just that valley and comes up with a solution that appears to be partly solving the problem, but not all of it. And a theme here, I think, is that it's actually quite hard to put together what I would call the HTE line of thought together with the precision medicine line of thought together with patient consumerism and choice. And I guess that's why it'll be our most important meeting, is that by the end of the day, we will have resolved these tensions. So I'm going to start with this piece from Richard Kravitz, who I guess might be on the line. So this is a lovely thought piece on what is heterogeneity of treatment effect. And in that piece, they articulate that there's sort of four broad strokes to it. One is this baseline probability of incurring a disease-related event. The other is responsiveness to treatment, vulnerability to adverse events, and utilities expressed by patients and maybe by paternalistic societies on different outcomes. And of course, when you read it, for all its elegance, you can realize that many of these points sort of further divide into others. And indeed, I would say that there are many axes of angst around heterogeneity of treatment effect. I would further say that the line of papers, going back to Joe Law and John Ioannidis' papers and David Kent's papers, and of course Peter Rothwell's papers and Rod's papers, I would call these the HTE papers. And whenever they tackle the problem, as they go down their valley, they make some assumptions. So you already heard from David that we'd like to predict response to outcome, but we're going to just predict risk of having the disease-related event. And largely, their papers have focused on the first and the third bullets. Baseline probability of incurring disease, coupled with instances where, for example, there may be a constant or flat risk to a drug-induced toxicity. At the same time, the rise of precision medicine has really focused, it's come very mechanistically. It's come from people who feel like they understand the disease on the inside, and so they've tended to focus on response to treatment. And then, of course, there's a whole field about utilities for different outcomes, which was alluded to at the end of the last talk, which I will ignore, not because it's important, but because I had to make some cuts to the talk. So let's just start by quickly covering some of the elements in RCT design coming from the beautiful studies from Rod and David and others. So it goes back to this classic first study, Peter Rothwell's study. Now, he's English, but he was in Edinburgh. And it's important to know that Archie Cochrane is from Glasgow. And so I think this assault on EBM was really just an old adage of Edinburgh trying to get one over on Glasgow. But of course, this was the analysis of the CAST studies and others that noticed that although there was an average effect, there appeared to be a very different accrued effect to different subgroups of patients. And importantly, in this very nice thought piece by David and Rod, they also point out that what you just heard about, that the patients at risk enrolled in these trials tend to be very left shifted, with most people that are relatively low risk. And certainly in my own field of critical care, on the right-hand side, you can see that's absolutely the case. When we admit people to the ICU, most of the people have a relatively low risk of dying, etc., even though we think of it as a far higher and more dangerous place than everywhere in the rest of the hospital. Many of the patients actually have a relatively low risk of dying. And so then in this same thought piece that I was sure David would already have shown, they beautifully unpicked that under situations where you have this median sitting to the left of the mean, where most people have a relatively low risk, the peril, I don't know if I have a pointer here, maybe I do. Yeah, the peril of the so-called one variable at a time subgroup analysis, men versus women, old versus young, etc., is that under situations with a left shift and a flat hazard from the intervention, then you get this situation where even if there's a large difference in the pale blue lines, the odds ratio for the overall effect for, say, age is 2. Like there's a big difference between the men and the women. That doesn't span far enough to the left and right of the mean estimate, and so you fail to notice that many of the patients to the left could actually be experiencing harm, and so therefore you want to have this multivariate risk model that spans across the entire range where you can have quantiles far to the left that would show up a robust estimate of, oops, this could actually be harmful, even though it was working very well on the far right. Okay, so I was glad when I read that because we had almost by accident decided we had to do this in sepsis with Zygris or Drotrecogin alfa. The original trial was in the New England, but at the time we had a hunch that it would be weird that all patients with sepsis would benefit equally from Drotrecogin, and all the subgroup analyses said, oh yes, there's no evidence of heterogeneity. So we built a multi-attribute risk model basically in the placebo arm and then generated a predicted risk of death and then an observed risk of death using the placebo control arm model to predict for quantiles moving from left to right in both arms. And early on in the very first trial, noticed that all the benefit was accrued by just a small number of patients on the right-hand side. Most of the patients were accrued in the left-hand side of the trial, and 5 years later, the drug was pulled from the market because none of the other trials could replicate it. So that's fine. And of course, then the answer would be we would just simply have to have huge sample size all the time, enrolling across the entire breadth of the disease of interest so that we always have enough sample to build these models. And so the answer to trialists is just to do huge, big trials and enrolling everyone at risk. Okay. At the same time, we've had this movement coming from precision medicine, which isn't thinking quite the same way. So if we think about if we go back to, for example, FDA parlance about predictive biomarkers versus prognostic biomarkers, if we were to start with the HTE world, we go back to this x-axis where we're taking something like risk of a disease-related event. That's essentially the risk that you would die from the disease, for example. If the primary outcome is short-term mortality, and you have all of that on the x-axis, it's not true that every intervention will have some sort of benefit that is in any way necessarily proportional to the increasing risk of the disease. It is true that if you're on the far left, you basically can't have any chance of getting the disease outcome, then you don't have any chance of getting the benefit. But as you move to the right, you may actually find that that's not exactly the same case. So for example, you can have a biomarker of disease that has nothing to do with whether the particular therapy would predict the outcome. And so that's why we have examples such as this mutation, which is actually associated with people having milder asthma, but more likely to benefit from a particular intervention. And then, of course, you get all these cross-dressers, which confuses the conversation because you have biomarkers that sort of look like they're prognostic and sort of predictive. And so people get confused in even keeping the terms in their head. And so, for example, in our field, we might have something like hypoxia. So hypoxia in the ICU is like a bad thing. If you have really a lot of hypoxia, then it's going to be hard to survive. And so we know that hypoxia is strongly associated with worse cases of ARDS or worse cases of acute respiratory failure, and it may or may not be associated with more likely to benefit from some interventions, even if not from all. So this isn't just true for a single biomarker. We can absolutely think about, in the world of precision medicine, having these sort of multi-channel markers to try to find patients. It could be a whole set of genomic markers, gene expression markers, underlying genetic variation, or it could be something like this where you don't need to read the x-axis. Suffice it to say, this was some work on some NHLBI trials where they looked at everything that was being measured at baseline, including all sorts of biomarkers and so-called sort of below the surface markers as well as clinical markers. And then use that basically doing a principal component analysis to divide into two phenotypes. So it sort of looks like multi-attribute risk, but it's only considering many variables, but then it bins everyone into just two groups. So it's actually just like the one-at-a-time variable analysis. Even though it used many variables, it divided just to, are you in phenotype 1 or phenotype 2? Now, they were very happy with themselves because these phenotypes weren't obviously predicted clinically, which is the data on the right. But then when they went on to see these otherwise invisible phenotypes, see what the implication were in the trials, in both the development trial and the validation cohort trial, the ARMA trial and the alveoli trial, they found that if you had phenotype 2, you were much more likely to die. At the same time, and so that's a prognostic phenotyping strategy, at the same time, the same phenotype in the second trial was highly predictive of benefit versus harm when exposed to the different strategies. And so then they ended up concluding that this may actually also be a predictive biomarker of change in outcome with therapy. So this is the essence of much of the precision medicine world, is trying to get at these notion of predictive biomarkers, but they just seem to have forgotten about this lesson learned from HTE about, there's a peril of having just a single subgrouping because it doesn't span across the market. There's also another issue that comes up here, and that is that the difference in effect size here isn't just a difference in the same direction, it's actually swinging from benefit to harm. Now, in the HTE papers, harm was usually seen as something, for example, expressed constantly, a drug toxicity. But that's not exactly what is at play here. So let's just talk a little bit about harm. So in my field, such as sepsis, we have a condition that we think is life-threatening organ dysfunction caused by a dysregulated host response to infection. And we think that that host response to infection is incredibly complex, with all sorts of complicated immune activation, with lots of yin and yang, lots of both pro-inflammatory elements and anti-inflammatory elements as the host is engaged in this dance with the pathogen. Ruslan Medzitov has this lovely conceptual model, and I apologize to Picori that the host is a mouse. But the purpose of this slide is simply to show that even that these axes of illness include both sort of green and red arrows. As the illness is playing out, there's a number of different host characteristics that can actually be competing against each other in this struggle to try to avoid one outcome, death. So you're trying to lower the edge of outcome, but in the disease process, before we've added any interventions, there's many ways of getting to that outcome. So in a mathematical model that tries to build up how do you die from sepsis? This group ended up trying to build a model using a whole bunch of PDEs, etc., but just trying to model from the underlying immunology why people get organ dysfunction, why they die. And then you can see here that if you just were to vary things that we never measure but we know vary, things like the pathogen load, the virulence, underlying genetic TNF response or anti-inflammatory response, then you get across very plausible ranges. This is a complicated slide, but the white are the people who are helped by treatment, and the black are the people who are killed by treatment, and the people in the middle, their fate is unchanged. And the point of this slide is just that it's all over the map, just by a different enrolled population and by ways that would not be predictable from a risk model or even from things that we commonly measure. And so the problem here is that you think you're studying one disease, but you're not really. Okay. What's so funny? So what can be done? So in the last couple of minutes, how are precision medicine folks thinking about this? So they basically have what I would call the hope and pray models, which is that they think there's a complex disease, they maybe have some putative biomarkers, and they either ignore the biomarkers or they take a bet ahead of time and only enroll based on the biomarker. And I won't speak about those further. Or else there are these so-called spread the bet models, where you acknowledge you don't know everything about the intervention, and you also don't know everything about the disease, and you're going to try to learn as you go. And the best and most evolved version conclusion of these spread the bet models are these so-called adaptive platform trials that Scott and Don Berry wrote about a few years ago, and then Janet Woodcott and Lisa Lavange from the FDA wrote a thought piece on them in the New England a couple of years ago. They're focusing on a disease, not just a treatment, considering multiple interventions with perpetual enrollment, often based on Bayes' theorem and tailoring choices over time. So far, they've been largely focused on the pre-approval space with an emphasis on efficiency and trying to get away with small and sometimes very small sample sizes. And actually just trying to work out what you graduate from phase 2 to phase 3, but there are now ones funded to sort of go into the comparative effectiveness space. The poster child is I-SPY 2. The first results came out about 18 months ago with 2 papers in the New England. And the randomization scheme looks like this. At the heart of it, this is something called response adaptive randomization. And just to quickly understand response adaptive randomization, if you did a regular trial and you were doing 400 patients, if you were 40 in and arm A was looking much better than B, and you were looking under the hood and you knew this, and you don't need to be the investigators, it can be a preset algorithm, then you take a bet on what to do with the next 40 patients. At this point, A is better than B is 78% certain, so now you could have the dice, or the die, when you roll it, 78 out of 100 times could actually come up A. So it's still random, but it's not 50-50. And so if the bet was right, then at 80 patients in, you're highly likely to be significant. You can already have set how you're going to control alpha error, and you can get out of the study early. The caveats, of course, is that if the second 40 was flat, you move back to the middle. It's also true that this is not very efficient for a 2-arm trial because you're still penalized by the smaller group. But it actually becomes very interesting under situations where we have multiple arms and multiple subgroups, which is arguably the situation we're in today. Heterogeneity at its heart is that we believe there are multiple subgroups, and comparative effectiveness is at its heart that there are multiple treatment options. So the heart of RAR is to think about having multiple options, and as the data accrue, they pre-populate a pre-specified Bayesian model that updates with posterior probabilities and then changes the randomization rule so that you could de-weight C and increase B, and you can have across different subgroups different weights for different kinds of patients being enrolled in the trial. And so I-SPY 2, I'm going to be done in a second. I feel like I'm running a little long, but a couple of slides. So I-SPY 2 took just 3 biomarkers, mammoprint, which is a prognostic marker of survival, whether you were hormone receptor positive or not, and whether you were HER2 positive or not. So there's 255 versions of this. It looks like 8 boxes. But actually, it can be any combination of sets of boxes. It's not 256 because one of them is that nothing works. So you take that off the list, but there are 255 ways in which any drug could be helping some subset of these people just with the 3 biomarkers. And so Don Berry had to sit the investigators in a room and say, you're not getting 255 cuts at the data. So he held them down to 10 biologically plausible subsets of these. But then they could expose patients across many of these subsets and then choose and narrow over time. And that's exactly what they did with the first 2 options. So they began the trial with uncertainty about which drugs works and in whom. And then they had preset thresholds for declaring, for example, that this particular intervention worked in this combination, so-called HER2 positive, HER negative, while at the same time this other intervention only worked in so-called triple negative. All right, so I left a lot out. But in conclusion, I think there are multiple axes of heterogeneity, and one of the challenges for this group is actually trying to have an all-encompassing summary of it all, not being trammeled by the fact that then when we get into the guts of trying to fix it, we hold some of the other problems constant while only fixing on one, and then we lose the audience that happen to be holding another problem constant. And in general, we've typically considered 1 or maybe 2 at a time. Classic HTE has largely focused on baseline risk of disease balanced against constant threat, and the peril being the 1-at-a-time subgroups, and the solution is large representative trials with post-trial multivariable model. Precision medicine designs didn't get the memo. The precision medicine people haven't been reading Kent and Hayward, and they've largely ignored the 1-at-a-time warning instead of focusing on these predictive markers which may not actually predict. They've been using these trial designs for putative predictive enrichment, of which hope and pray are really a hope and a prayer. But spread the bet is quite exciting. It's working in cancer, and it's also arguably more patient-centered in that even in the middle of accruing the information, as quickly as it's possible, you de-risk the exposure to patients. And so you're actually in the opposite direction of what we need is this huge trial so that we can then understand after the fact who it works in. Now, it's not clear that it's patient-centered, even if it's convenient, it might not actually be patient-centered. With that, I'll stop.